Hello everyone and welcome back. Okay, so in the previous video we talked about how the neuron works as kind of like a miniature factory. So we talked about all the components of a cell, such as membrane, nuclear membrane, nucleus, ER, Golgi apparatus, mitochondria, lysosomes, tubules, and microfilaments, and how the membrane works. And then in this video, we will talk about the production of proteins. So we know that proteins are a product of our little cell factories, but how are they made? So in this video, we will talk about chromosomes, DNA, genes, amino acids, and then the whole process of protein synthesis through transcription and translation. Okay, so we talked about the cell being a protein factory in the previous video, so let's keep going with that. So our nucleus would technically be the CEO of this miniature factory. And the nucleus would contain all the blueprints for these proteins, so all the instructions. So these blueprints are stored in here, so in the nucleus, and then they will have to be copied, and then they will be sent to the factory floor to technically be folded up in certain structures. So these blueprints specifically are the genes. Genes are small segments of DNA, and DNA rolls up into a chromosome. So here in this picture, it's pretty clear. So here you have a cell, here you have the nucleus. In the nucleus, you have all these chromosomes, which look like this. And then if you unwind those, eventually you'll get DNA. And a part of the DNA would be a gene. So the DNA is a double helical structure. And if you cut this part off, that is the gene. So DNA consists of 4 nucleotide bases. So here you can see the 4 different types. You have adenine right here and thymine right here. So in further lectures, I'm just going to talk about A and T. And then you have guanine right here and cytosine. So G and C. So A and T always connect to one another, and C and G always connect to one another. G cannot connect to A or T, etc., etc. Okay, so here you have a little recap of what I just talked about, but then in the form of the neuron. So here you have the neuron, here you have the dendrites, dendritic spines, here you have the nucleus, and inside the nucleus you have a lot of chromosomes. So these are rolled up pieces of these double helix or double-stranded structures with their nucleotide bases. So again, A binds to T, or adenine binds to thymine, and G binds to C, so guanine binds to cytosine. Okay, so within the chromosomes, sequences of hundreds to thousands of nucleotide bases spell out our genetic code. So this genetic code, technically it codes the order of which amino acids should be assembled. So it's technically a list of instructions of how these proteins or amino acids will be assembled. So I know I talk about amino acids and proteins interchangeably, but they're technically not the same. Amino acids are the building blocks of proteins. So if you have a bunch of amino acids right here, and if you connect them all together in a polypeptide chain, and then if you would fold those up, you eventually will have a protein. So obviously this is very, very simplified, but we will talk about that a little bit more in a later slide. Okay, so to initiate the protein synthesis, a gene segment of the DNA will unwind. So the two sides of the DNA will technically just go apart. Not the entire DNA, but just a part of it. And then the exposed sequence of nucleotide bases on one DNA strand will technically serve as the template to attract nucleotide bases that are free-floating inside of the cell. So these nucleotides will attach to the template strand and create a copy of the strand called RNA, or ribonucleic acid. So technically what it does, DNA unwinds, or a part of the DNA unwinds, then free-floating nucleotides, so the ATCGs, they will attach to one strand, to the template strand, and they will create a copy, and this copy is called the ribonucleic acid, or RNA. So this single strand of copied information from that gene is technically just a copy. So this whole process is called transcription. So later on we're going to talk about translation. So transcription is just making a copy of a part of the DNA. The only difference between RNA and DNA is that DNA has the nucleotide called, RNA has the nucleotide called uracil instead of thymine. So instead of T, RNA will have a U. So the transcribed strand of RNA is called the messenger RNA because it has all the genetic code in it, and eventually it will go out of the nucleus and it will go to the endoplasmic reticulum, where these proteins eventually will be made. Okay, so here is what I just said in one picture. So here you have the DNA. The DNA is all like coiled up, but if a part of the DNA will unwind and these will be separated from one another, in this case, the top one will be the template strand. So free-floating items, free-floating nucleotide bases will attach to these little parts. And then eventually they will form the RNA, or the ribonucleic acid. So as you can see, instead of the T's right here, RNA has U's. So this should be an exact copy of this, except for the T's are U's. So it's called messenger RNA because it sends this genetic code to the outside of the nucleus, to the endoplasmic reticulum. So when this messenger RNA gets to the endoplasmic reticulum, the protein eventually will be created here. So how is it created? So on the endoplasmatic reticulum, there are proteins called ribosomes. And ribosomes act as catalysts to build the protein. So they are the protein builders. So when the messenger RNA reaches the endoplasmic reticulum, it passes through a ribosome, and this ribosome will read this genetic code. And technically, this reading of the genetic code is called translation. And during this process, a particular sequence of nucleotide bases in the messenger RNA are transformed into amino acid sequences. So again, once this RNA is formed, it will go to the ER, to the endoplasmic reticulum. It will pass through a ribosome, and here the ribosome will technically read what's in the genetic code and it will translate it into amino acid language. So when multiple amino acids are chained together, they form a polypeptide chain, and then a combination of more polypeptide chains will form a complex protein. Okay, so I know there was a lot, so let's go over it through the pictures. So let's first go over a more simplified picture. So you have DNA, it unwinds, so it's not all curled up, it unwinds. Then these two are getting separated. In this case, the lower one will be the template strand of the DNA. Then free-floating nucleotide bases will attach to these to the template strand, and that will form the RNA. So this whole process is called transcription. So then you have the messenger RNA. It goes outside of the nucleus and it goes into the endoplasmic reticulum. And there, within a ribosome, it is translated into a polypeptide chain. So into all these amino acids. So a ribosome is technically a translator. So let's say the messenger RNA language is Chinese and the polypeptide chain is English. So the ribosome is just translating the Chinese into English. So let's look at this picture. So again, in the beginning, the DNA uncoils to expose a gene, which is a sequence of nucleotide bases that encodes a protein. One strand of the gene serves as a template for transcribing a molecule for messenger RNA. The messenger RNA will leave the nucleus and comes in contact with the ribosome in the ER. And then as the ribosome moves along the messenger RNA, it translates the bases into specific amino acid chain, which will eventually form this protein. Okay, so here I have it all together. So from DNA to messenger RNA, aka you're just making a copy, this is called transcription. And from messenger RNA to protein is called translation. Okay, so long polypeptide chains have a tendency to curl up into either a helix or they form what's called pleated sheets. So these shapes are called secondary structures. So the primary structure is just a polypeptide chain, and the secondary structure is a helix or a pleated sheet of these polypeptide chains. So then these structures in turn also like to fold together and make a more complex shape. So this is what we call a tertiary structure. And when these polypeptide chains are in their tertiary structure, it is technically already a protein. So it's a very simple protein. And if you combine more of these tertiary structures, you can combine a more complex protein. So here you see that here you have a polypeptide chain, it makes a pleated strand or a helix. So there's a secondary structure. Then if that coils up even more, it's tertiary structure. And you can get what's called a quaternary structure or a more complex protein by putting more tertiary structure proteins together. Okay, so we're almost there. So after these proteins are created, they get sent to the Golgi bodies, or Golgi system. We already talked about this. So here they're just put in a pretty box and labeled. And then they will be loaded onto motor molecules, onto the microtubules, and then they will go to their final destination. So here's a little recap. This is the last slide. So proteins, the blueprint comes from within the nucleus. Then proteins will be formed on the endoplasmic reticulum through the ribosomes. Then they will enter the Golgi system where they are wrapped in a membrane or put in a nice box with a bow on it. And they are given a shipping address or shipping label. Then each protein package is attached to a motor molecule and will move along the microtubule to its destination. So then after this protein is created, a bunch of things can happen to it. So wherever proteins are needed, these proteins will be created and will be sent to their destination. So here are some examples. A protein may be incorporated into the membrane. It can become a receptor. It can also act as an enzyme, so it can stay within the cell. Or it can be sent outside of the cell for other functions. And this was the end of the video. I will see you guys back soon in the next video.